Now, I'd just like to introduce my colleague, Dave Chandler. He's going to talk about biopesticide application, a series of interconnected challenges. Dave led AHDB Horticulture's AMBER project, which was to identify practical ways for growers to improve the performance of these products in their crop protection programs. So over to you, Dave. You might have to unmute yourself.
Excellent. Is that working, Rosemary?
Yes, yeah, great. Thank you.
Brilliant. Okay, so good afternoon, everybody. It's really good to see a kind of really good turnout. So many people coming to listen to the talk. So I'm going to talk about biopesticide application in a kind of a broad context, and that's why I've entitled this talk kind of a series of interconnected challenges. And it's really about using biopesticides as part of integrated pest management. So this work, the work I'm going to present, it is a team effort. So you can see here that the people that's involved, and at the very end I'll do a big list of everyone who's involved in the research that we've done over the years. So a lot of the work that you will see here was done by my colleague Jill Prince. A key person actually in biopesticide application is Claire Butler-Ellis at Silso Spray Applications Unit, and Silso are world leaders in all things to do with pesticide application, and Claire has really pioneered a lot of our work on biopesticides. I think she may be joining us this afternoon, so she may, if she is able to join us, she can help with some of the questions we'll get inevitably. So Jude Bennison at ADAS, Tom and Joe at Harper, and Toby Roos at Kiel have all been involved in our work on biopesticides and IPM. And then we have two consultants, Roma Gwynn, who's a specialist in biopesticides, and Rob Jacobson, who's a specialist in all things integrated pest management and biopesticides for protected crops. So everything that I'll present now is based on the work and the collective view of these people, and I cannot thank them enough. Okay, so I guess this slide is really to start us off, and it's all about how biopesticides really fit in with integrated pest management as part of the green transition that we urgently and desperately need for a sustainable farming system. So as I'm sure you're all very aware, the availability of conventional synthetic chemical pesticides is declining. You can see this bar graph here at the bottom on the right. This just shows the availability of pesticides within the European Union as active substances. And the fall in a decade from 1998 to 2008. So in 98, there were about 800, 900 active substances registered in the EU, and within a decade, because of policy initiatives and other things, it had fallen to 400, and it's pretty much stayed about 400 ever since then. And we know that we need, I don't need to teach you any of this, but we need sustainable systems for controlling pests, and by pests I mean invertebrates, plant diseases and weeds. We know that pests are a major constraint on the production of crops for food and for other uses, and we need to transition to a more sustainable system of managing them. And the way to achieve that sustainability is through integrated pest management. And on the left here, you can see an IPM pyramid. This is from one of Rosemary's papers talking about IPM in vegetable crops. And again, I'm sure you're very familiar with IPM and the pyramid concept, in which you layer different systems of IPM, starting with prevention, followed by detection systems, and on the top you have control. And the idea of the pyramid is that it's kind of like a cake. It's also like mutually reinforcing. So things you do for prevention and for decision support mean that you should be less reliant on control measures. And then with control measures, you should start with mechanical, physical, and kind of natural controls, followed by biological controls. And then you're using chemicals including biological pesticides, but also synthetic chemistry, really as a top layer, as a last resort. So you're using these things as a precious resource. And this is what we want to achieve for IPM, and this is the way forward. The issues, of course, is that growers and farmers need to have enough tools in the IPM toolkit to enable them to achieve safe, effective and affordable pest management. And that isn't the case at the moment, because we're seeing reductions in the availability of synthetic chemistry, and we're not getting enough alternative approaches in the hands of farmers and growers. So we're going to talk about biopesticides. So biopesticides should be, sorry about the spelling mistake, biopesticides should be seen as part of this drive to promote alternative approaches in IPM, which will include the adoption of biopesticides. They're also known as bioprotectants. What are they? Well, they are plant protection products that are based on living microorganisms and natural products. And this is an important point, because biopesticides encompass a very wide range of entities now. So we have the living microorganisms, so bacteria, fungi, viruses, but we also have natural products such as peptides, RNA for RNA interference, things like terpenes from plant extract, ketones, fatty acids, etc., etc. So we actually have a wide range of entities, and of course all these different entities, which are classed as biopesticides, all have their own particular characteristics, their own particular use requirements, and that means that you need to have a lot of knowledge and more attention to detail to apply to each individual case in order to get the best out of these products. This thing on the right, this is an example. This is an insect pathogenic fungus, Bavaria bassiana, which is infected and killed, in this case, a caterpillar pest. The fungus produces spores, which have got the ability to adhere to and germinate on insect cuticle. And then if it's sprayed onto insects, the spores enter the insects, they penetrate it, they overcome the insect immune system, they kill the insect. And then after death, the fungus grows out through the body, and you can see all the spores produced on the fungal hyphal here, and this can spread to other insects. So it's a really good example of a microorganism as a biological pesticide. When I first started working here at Wellesbourne over 30 years ago now, kind of biological pesticides were seen as something as a niche, but they're not. They're in the mainstream now. So in the UK, for example, we have 50 biopesticide or bioprotectant active substances registered and approved for use, and that's actually 17% of the total number of active substances approved in the UK, and that number's going to go up. It's 8% in the EU, but those numbers are going to go up in the next years and decades, so that these substances are very much part of the mainstream. And I think although synthetic chemical pesticides will be a mainstay of crop protection moving forward, I think we'll see the biopesticides play an increasing role. Put this bullet point in bold here, because this is a thing I want to focus on for this webinar, is that the thing that unites these biopesticides is that they're sprayable, they're formulated. Many of them, in fact most of them, are contact acting, so they're non-systemic, and that is really important in terms of spray application, because I think we've had a luxury with synthetic chemical pesticides in the past, in that when you've got synthetic chemicals that are systemic, you can actually get away with poor application if you don't directly hit the target, but as long as you hit the plant, you'll still get effective pest control. But with biopesticides, particularly the contact acting ones, which, as I said, the majority are contact acting, then you've really got to be on the top of your game when it comes to application. And also remember that these biopesticide products, they often have a lower intrinsic potency than synthetic chemical pesticides. So it's really about kind of winning, I call it winning the numbers game, which is about delivering an effective dose at the right time and the right place in order to capture their benefits and get effective control of target pests. The other point I wanted to make on this slide is that this is about where the innovation is coming from. So 80% of biopesticide companies are small to medium and enterprises, and this is important. It means that the innovation is coming from these SMEs, but SMEs, although they're often lean and flexible companies, they lack the resources, the funds to put into things like pesticide application. They tend to focus on development of a product and the product development pipeline and getting a product towards the authorization stage, and they probably don't have the resources that they would really like to focus on the application stage. And that's something where I think we as a kind of a research community and farmers and growers and people in industry need to work together on to pool our collective strengths so we can address on application. And I might, if I've got time, I'll come back to that towards the end. Okay, so this slide is just some examples of different biopesticides. So I want to emphasize, I'm not selling product, I'm not advocating use of any particular product in this slide. I'm just kind of showing examples of them. So here we have biofungicides. The example I've given here is Bacillus aeroliquefaciens. One of the products that's available based on Bacillus aeroliquefaciens is the product Serenade, which is produced by Bayer. So this is a bacterium which can be a very effective biofungicide. It produces metabolites which have got antifungal action, and it can work really, really well. But there are, of course, other biofungicide products in a development pipeline and which are on the market. We also have bioherbicides. The example I've given here is pelargonic acid, and there's a product called Catown Gold, which is produced by Certin. So that's a kind of a broad spectrum bioherbicide. There's other products here. I put this one, Firehawk, which is based on nonanoic acid, which is sold and is available in Australia. And the reason I've put that is that if you look for that product, there's a really interesting bit on their website where they show application at field scale for this particular product. And one of the concerns people have always had in the history of biopesticides is, can you actually produce enough of them, and can you actually develop them at commercial large scales in fields? And this link kind of shows that it's possible. And then the last of the three groups are the bioinsecticides, and probably the best known bioinsecticides are those based on the bacterium Bacillus thuringiensis. This is an electron micrograph of Bacillus thuringiensis here. This is a bacterium that, as part of its life cycle, produces a spore. You can see that in the bottom right hand corner of the micrograph, but in the top left you can see this structure here. This is a protein crystal which has got insecticidal properties. This works by after it's ingested, it binds to the gut to specific receptors in the target insect and causes gut leakage. This is used in this country, for example, against Lepidoptera larvae. So there's Project Delphin from Andermatt. And as I said, there are other products available in the UK and Europe that based on the same kind of actives and against the same range of pests. This is relevant, and I put this in the other day. So the UK, through DEFRA, the Department for Environment, Food and Rural Affairs, on Friday they launched their National Action Plan for Pesticides in 2025, and it's available on the web. I put the link at the bottom here, and I would encourage everyone to look at it. And it's linked quite closely to the EU's drive as well for sustainable use of pesticides. So the National Action Plan from DEFRA is all about integrated pest management, as we saw in Rosemary's IPM pyramid slide earlier. So I've just taken this as the quote. This is from the kind of the top of the strategy. I'll just read it out for you. It says, so the strategy is all about I want to encourage the development and uptake of IPM and alternative approaches or techniques to reduce dependency on the use of pesticides. And that's twofold, my interpretation of that is twofold. One is to make crop protection safer for people and the environment, but also to make it more effective and sustainable, because by using IPM, you in principle reduce the selection pressure for the evolution of resistance to pesticides in target pest populations. And if you can reduce resistance evolving, then you can continue to use your synthetic chemical pesticide as a precious resource. So that's a kind of the broad overview, and within the National Action Plan, there are targets being set out for the reduction of risks and impacts of pesticide use, including pesticide active substances of particular concern because of perceived issues around their safety to people and to the broader environment. It also includes horizon scanning for to identify pest control gaps. And that's really important. In fact, I would say we shouldn't be horizon scanning. We should be working in that area as a matter of urgency, because our farmers and growers, particularly in this country, don't have enough plant protection products to enable them to control all pests all of the time. So there's a matter of urgency there. And biopesticides come into the National Action Plan. So they form part of the action-based indicators that DEFRA have developed and adopted, which includes the broad uptake of IPM, the availability of biopesticides, and things like training of professional plant protection product users. And DEFRA have also said that they will continue support for biopesticides applications through the registration process. Okay, so we've talked about what biopesticides are, and we've looked at kind of broad range of products that are available. This slide just looks at some of the benefits and costs of them, and this is important because what we need to do in IPM is capture the benefits and mitigate for the costs. So the big benefit is really their human and environmental safety, and the fact that many of them don't produce toxic residue. Many biopesticides are specific, so again that has benefits in terms of not having harmful effects on non-target organisms. The things that are alive, remember that picture of that white fungus Bavaria bassiana infecting the caterpillar that I showed earlier. So that shows the potential to give an element of self-sustaining control. So with the microbial biopesticides, they tend to be applied according to an inundation strategy, in which you apply a large amount of the microbial control agent, certainly larger than occurs naturally in agroecosystems, and you expect pest control to be done by the material that you actually spray onto the target crop for the target pest or disease or weed. But you may get some added value by the microorganism persisting in the environment for a certain length of time as it reproduces. And what will happen over time is that the population will die back to the carrying capacity of the ecosystem. So you get this element of self-sustaining control. They also, certainly the living microbes have multi-component activity, which I'll show in, I think, in one of the next slides. So they can, for example, be involved in plant growth promotion, and they can be applied with conventional spray equipment, which is a benefit because they can be slotted into existing crop protection practices. The downsides is that many of these products have lower inherent potency. I put a question mark there because that's not always the case, and certainly I think with next generation products that will come through, they'll see things with higher potency. They tend to be less robust and less forgiving. For example, they tend to have lower persistence in environment compared to synthetic conventional chemical pesticides. So that can be a downside in terms of persistence of effect, but of course it's a benefit in terms of lower negative impact on the environment. So we have to weigh up these things because obviously it's all about kind of balancing there. because they're natural products, and so the natural products are subject to naturally occurring degradation pathways in agroecosystems, and the microorganisms themselves, as I said, tend to be reduced quite quickly down to the natural carrying capacity of the environment. They have lower persistence, and because the microbes are alive, they're going to be affected by environmental conditions like temperature, like ultraviolet light, impact of sunlight and things like that. And, you know, I said this at the beginning, I said that we have a wide range of entities and they've all got their own specific requirements, which means that they need greater knowledge and attention to detail compared to old style chemistry for synthetic pesticides. That's not a problem because, you know, we've got very capable, informed growers and farmers and end users, but it does mean we have to put effort into education and effort into ensuring that end users really give that attention to detail to get the best out of these products. And cost is an issue, so they tend to be more expensive at the moment compared to conventionals, but those costs will come down as we get scale of use. Okay, so this is the point I made earlier about bioprotectants having multiple modes of action, and this is an example for trichoderma, which is used against soil-borne pathogens, but many of these statements apply to a lot of the microbial biofungicides. So, for example, they can produce antimicrobial compounds that kill plant pathogens. They can compete with plant pathogens for space. They interact with the plant by turning on plant defenses against diseases. Some of them are mycoparasitic, so they invade and kill plant pathogens. They tend to be best at low to moderate disease levels. I guess the point about this is that when you've got multiple modes of action, then that reduces, in principle, the probability of resistance occurring, which is really important. And you can get this added value in terms of better quality plants. In fact, we see the same thing also for some of the bioinsecticides. So I work a lot with insect pathogenic fungi, for example, and we know that they interact with plants, they can grow endophytically, they can turn on plant defenses, they can also boost plant growth. So really we need to be able to using these biopesticides in ways that actually capture these attractive biologically based properties to give this added value in crop protection. Okay, so I'm going to talk about now, move on to kind of use of biopesticides, and then I'll focus in about 5 minutes specifically on issues to do with their application. So this slide is about biofungicides, and there's very strong evidence from the research literature that biofungicides can work well in spray programs. There's strong evidence that they can be really useful when you incorporate them in a rotation. So you're substituting some of your synthetic chemical fungicides for a biofungicide, and that's well established. There's also evidence that you can actually get very effective control when you use a bio-only program. And I've got an example of that here. So this is work that Angela Berry and Zhang Mingzhu did Naib East Malling on control of powdery mildew on strawberry, and these graphs here are comparing the effect of different treatments on. You can see this top graph is number of mildewed fruit, this bottom graph is percentage of mildew fruit, and they had different treatments. They had an untreated set of conditions, and then they had a conventional fungicide spray, then they had application of a biofungicide based on a bacterium, Bacillus pumilis, and then application of a biofungicide with a biostimulant, and also application of the biofungicide with silicon, which can also have effects and efficacy against powdery mildew. And I think the point about this slide is, look at this, all those different treatments, the biological treatments and the fungicide treatments, all had effectively the same level of efficacy. So you can get, in principle, the same level of control with biofungicides that you can with a conventional fungicide program. Okay, but that's on an experimental scale. And the key question is whether you can and how you can actually translate that knowledge and information from an experimental scale to a commercial scale for commercial farming, when you're having to deal with variable environmental conditions, where farmers and growers are naturally risk averse because there's little room for error and so on. And that's, I think, a big challenge for us moving forward, and something we need to address. There are also issues about if you are combining biofungicides and chemical fungicides in a spray program, you have to make sure that things are compatible, and you have to do it as part of an integrated pest management system. And in this case, the way forward is to target your applications in a precision spray system by combining applications with disease forecasting. And I mentioned this before, and it's all about, getting high quality produce, reducing the disease population below that action threshold, and by reducing your reliance on synthetic chemical fungicides, you're reducing the resistance pressure. So can you use these things at scale? I think you can. This is an example from the Crop Health North project. This is actually featured in the National Action Plan on the DEFRA website. And this was work that was done by the Yorkshire Agricultural Society with people you can see here on the right. So Roma led a lot of it through her work as a biopesticide consultant with Rationale. And there's also a lot of work with Newcastle and Eurofins and Stockbridge Technology Centre. So this was GEP standard trials done at 3 sites in the northeast of England, focusing on disease control in spring wheat and winter wheat, and looking at things like fusarium, yellow rust, powdery mildew, and septorium. And they compared 3 regimes. So a biological regime, which combined microbial seed treatments with botanical and microbial foliage sprays, which were applied at risk periods. So this was targeted sprays, not calendar sprays. This was targeted sprays. I can't tell you what those products are. In fact, I don't actually know what they are because they were all off label. So these are coded products. The second regime was an IPM regime, in which conventional fungicides were applied during at-risk periods. And then the third regime was a standard chemical regime, in which you had conventional fungicide sprays applied at standard timings for crop physiological age. And I'll show you some of the results. So this is when the trials for the second year of the Crop Health North project. And it's showing you the yields for winter wheat with 2 different varieties in replicated plots at 3 locations, so Stockbridge, Cockle Park and Nafferton. And the key thing here, look, we've got the 2 varieties for yields for our IPM system, that's targeted chemicals, our standard chemical fungicide spray on our biological system. And the key point here is that there was no significant differences between any of the management regimes or the sites in terms of yield. So it shows you that you can achieve the same yields using a biological system as you can for an IPM chemical system or a conventional calendar chemical spray, which I think is a major step forward. This is data from trials they did in the 3rd year of the study. Again, it's for winter wheat. It's showing yield. It's for 3 different locations for the 3 different treatments: the targeted chemical, the conventional chemical, and the targeted biological. And again, my take on this is that if you actually, sorry, we'll go back to it. If you actually look at the data, there aren't big differences in yield. There are some kind of the fine margins of the yield. So they're kind of implications that maybe the yields for the conventional is slightly higher than for the biological, but it's not big. And I think that's really important. It's proof of concept. It's showing that at scale, you can actually get effective crop protection using biologicals. This is for bioinsecticides. This is data that I did some time ago, and again, it's proof of concept. This is on tomato crops done here at Wellesbourne, and we were looking to see if we could substitute conventional acaricide for spider mite control with a biopesticide, and in this case, we were looking to substitute phenbutadiene oxide, which has now been banned, with a fungus, Bavaria bassiana, with the product Naturalis. And we were using it not as a standalone treatment, but as part of a supplemental treatment, a second line of defense, if you like, for spider mite control with Phytocelis predators. So they're inevitably, if you're doing IPM on spider mite control on tomatoes, there's inevitably times in the season where the pest population, the spider mite, would start to outstrip the ability of the predatory mite to control it, and that's when you want to come in with a fast-acting supplemental treatment. And it used to be the case that growers were using conventional pesticides for that. In fact, they still do, but we wanted to see if you could substitute that for this fungus. And so this is what the data on the graphs you can see on the right are for. So this is data for our control treatment. So we had basically large-scale glasshouse experiments here at Wellesbourne. So we had a control in which had spider mites infected on tomato. We applied phytomites, but we applied a low level of phytomites to simulate a situation in which we're getting lack of control, and we wanted to come in with a supplemental treatment. And the supplemental treatments were phenbutadiene oxide or a fungal treatment Naturalis. And the point about this is that we're getting really good control of spider mite, both adults, nymphs, and eggs, with the fungus. So it's a rare example, actually, I think, of the biopesticide actually outstripping and outperforming the ability of the chemical pesticide. So I'm just aware of time, but because I'm talking too much as usual, but it's a good example showing that these biopesticides can really work. The question is how you get the best out of them. So we've known through research that biopesticides can work well, but we know also that growers have reported sometimes mixed experiences in commercial practice. And this is where our AHDB Amber project came in, which we did some years ago. It's a project between Warwick, ADAS and Silso, where we were looking at how growers use biopesticides on protected edible, ornamental and hardy nursery stock crops. And the idea was to observe how growers use them and to identify reasons where and why biopesticides can sometimes perform suboptimally in commercial practice, so that we could develop mitigations to improve biopesticide performance in horticultural crops. So we evaluated biopesticides with growers who were very experienced growers but had not by and large used biopesticides before, and we observed how they use biopesticides on 7 crops: pepper crops, cucumbers and ornamentals, all done at commercial scale, so very large scale, against 3 pests, 3 diseases. We studied 3 bioinsecticides and 3 biofungicides. And I guess the upshot, these were the key lessons in relation to what I'm talking about today, is that first of all, we found that our growers need accessible information to optimize their use, particularly when it comes to application. Remember, I said at the beginning that you have to be on it application, and you need to deliver an effective dose at the right place and the right time. And growers really need information about what the effective dose is, what the persistence of the product is, the environmental conditions that are needed, and precise information about how to apply it to get best use of it. We need a more systematic approach to optimize application for spray application of these products. And we still lack now, precision tools to deliver the effective dose at the right time and the right place for these contact acting products. And this is data from Silso. So I'm going to talk about the next 3 or 4 slides, really get into the hard detail on application. So this is data that Silso generated in their amazing track sprayer facility, and they were using a tracer dye applied at a constant concentration. In this case, this particular graph you can see here it's showing the results applied to a low flat crop. In fact, I think this was particularly for an ornamental crop, but the same basic principles apply to things like basil as well. And what they were doing was that they're applying a constant concentration of a tracer dye, but at increasing water volumes. And you have two graphs here. I must apologize, I'm profoundly colorblind, so I'm not quite sure what the dark color is. I think it's gray or it could be blue. And this other one here going from top left to bottom right, I think that might be orange, but I'm not quite sure. Anyway, I'm going to call this one blue, right? It probably isn't blue, but this curve here shows the total deposit of tracer dye in terms of microliters of material per gram of leaf. Remember, it's constant concentration. As you see, as the volume goes up, more material is deposited on the leaf, but you get a plateau where you kind of reach the carrying capacity of the leaf, and that occurs at about 600 litres per hectare. That's important because at the time the label recommendations, and in fact it still applies for a lot of biopesticides, they go up to 1500 litres per hectare, and that water volume is really, really high. And about 1500 litres per hectare, you may well be getting runoff, which is a problem because you're going to be losing product. This line here is a normalized deposit. So this is the amount of material per gram of leaf per 100 litres per hectare of water. And I kind of view it as the efficiency, if you like, of production. You can see this is quite amazing because it shows you that it's going down really quite from the very, very beginning. And my kind of simplistic interpretation is that your sweet spot is about here, is about the kind of the 500 litres mark. So as I said, the label recs for horticultural crops often say up to 1500 litres with a constant dose application, but the concern is that the amount deposited on plants, and hence the efficacy, can be sensitive to water volume, certainly for short flat crops, as we've seen this example there. And then the upshot of that is if you're applying a lower water volume, you're not getting issues with runoff, you're not getting this plateau situation that we can see in the graph on the left, and also spraying is faster, so it's more efficient, and because you're not getting runoff, you're not gonna get the waste that you want to avoid. And the final point is that, biopesticides have an optimal water volume for efficacy, but we don't actually know what it is for most products. So that's something we need to find out in the future. So these are recommendations from Claire and her team at Silso for the short flat crops like herbs, like ornamentals, and the amount of active substance deposited on the plant was found to be sensitive to volume. And the implications are as follows. So for a product applied at a constant concentration, the maximum volume is about 1000 litres per hectare, but for smaller plants, we're saying it's better to go for 500 litres per hectare. So that's actually quite low. For a product applied at a constant dose, and in fact, most products are applied at a constant dose application system, then the maximum active substance is going to be achieved on the crop at the lowest water volume. But of course, you mustn't exceed the maximum label concentration. You must stay within the label recommendations. We also did work to a lesser degree, but with a vertical crop, in this case with tomatoes, which were applied tracer dye on a vertical boom. And we found in that case, the amount of active substance, in this case, a tracer dye, which is a proxy for active substance, was found to be relatively insensitive to water volume. And the reason for that is to do with the complex architecture of the crop and the layering of the leaves, and that's quite profound. The implications for that from the Silso team is that if you're applying a product at a constant concentration, then the maximum volume you want to get is like 1000 to 1500 litres per hectare based on the data that came from that study. For a constant dose application, then you're better off going for a low water volume within the label recommendations, simply because it's so much faster to spray, and therefore it makes application time efficient. Silso have also done it's really useful. They've done a calculator, because what the calculator does is it actually converts the volume needed for the vertical crop wall into the volume applied for the floor area, because of course this gets really complicated. The label recommendations in terms of dose are all about the floor area, but in fact what you're spraying is a vertical wall area. So the Silso calculator is really useful and kind of gets around that. So it ensures that the label recommendations aren't actually exceeded. Right. So this is data that we did in the Amber project that continued on from the stuff that kind of seen previously. And this was a track sprayer experiment using a fungal biopesticide, Bavaria bassiana, in a product called Botanigard, applied against spider mites on tomato. And it was done at a nominal constant dose. So we applied the fungal product at different water volumes, so 250, 500, 750, 1,000 and 1,500 litres per hectare. But it was done at a nominal constant dose. The idea was that the water volume increased, but actually the dose we want to deliver in terms of amount of fungus per unit area of leaf should have remained the same. And the graph is for the amount of fungus as spore counts per unit area of leaf against the different water volumes. For the top side of the leaf, you can see that blue colour, and then for the lower side of the leaf. So the first thing to notice is that the amount on the underside of the leaf is really, really small, and that's not surprising, because this is the contact acting product. It's not systemic. The second thing is that we were aiming for a constant dose, but we didn't get that. And in actual fact, the amount of material we applied was affected by the water volume. So we got the highest concentration of fungal spores on the leaves at 500 litres per hectare. And we don't know why that was the case. We need to do more work to find that out. Another point about that is that dose translated into efficacy. So these boxes here, these boxes are the survival time in days. So we got the shortest survival time for the highest dose. We got an average survival time of 3 days for 500 litres, and average survival time for 5 days where we got for these larger volumes. So I guess it's showing you that water volume can affect dose, and dose affects efficacy, but we don't know why water volume affected dose, and we need to find out more about that. But that's really important going forward. Another point on this slide is that you need to get the basic rights for biopesticide application, because you need to pay attention to detail. So you need to invest in your spray equipment, you need to do your calibration, you need to check your equipment, you need to change your nozzles, you need to think about compatibility between biopesticides and conventional pesticides. So we know some growers, for example, who use biopesticides, they have a separate biopesticide tank, so they're not tanks mixing with conventional chemical pesticides. Think about water hardness, think about use of adjuvants, think about how you're doing your tank cleaning and your pipe cleanings, because pipe cleaners can be harmful to biopesticides. And be aware that, the standard advice you get on training is to triple rinse, but triple rinsing may not be enough, for example, to wash all the residue out of a chemical pesticide, which may then have a knock-on negative effect on a biopesticide if used in the same tank. Right, I'm gonna finish off. I am aware I have run over time slightly, but I'm going to go very quickly over another point, which is that application isn't just delivering biopesticide to a target. I view it as part of an integrated pest management system. So yes, you want to have effective, efficient biopesticide delivery so that you can get the desired performance of your product against the target pest, but you need to use that with decision support. So you need to know when you're going to spray. You need to understand the effect of environmental conditions on your application, because environmental conditions can have a really influential impact on efficacy. You need to understand the effect of other control agents, and you need to understand how your biopesticide use fits in with other examples of agronomic practice, for example, crop genotype. And in fact, I'm going to focus on that for the next couple of slides. So this is work we did in a BBSRC project called a SARIC project, which was done between Warwick, Keele, Harper, Durham and ADAS. And we were looking at the interaction between host plant genotype and biopesticides for the control of aphids on brassica crops. Okay, and this slide here shows data from Toby Bruce and his team at Keele University, where they were studying the effect of a plant activator called cisjasmone, which was sprayed onto brassica plants. And cisjasmone, as I said, it's a plant activator. It turns on plant defense genes, and it also can make plants more attractive to natural enemies, including parasitoids. So this set of graphs here shows the effect of cisjasmone on parasitoid activity, measured as a number of mummified aphids, so aphids that have been parasitized by a parasitoid. With and without the cisjasmones, the grey is with cisjasmone, and this, I'm assuming it's orange colour here, is a control. And it's done for different types of brassica. And the key point about this is, can you see the little stars show where you've got a statistically significant effect? Sometimes the cisjasmone worked and it increased parasitoid activity against aphids, and sometimes it didn't. And the activity was dependent upon the plant type. In other words, what I'm saying is plant genotype can determine the success of this particular biopesticide operating as a plant activator. I hope I explained that properly. And I think that's something we've probably not considered fully up until now, is that the crop variety can have a significant impact on biopesticide efficacy, and we need to take that into account. This is about data for field trials. So this is again using the same compound, cisjasmone, and when you apply cisjasmone to brassica plants, if the plant is activated, the plant produces deterrent compounds which actually can deter aphids from landing on the plant. And again, this action of the cisjasmone is dependent upon plant genotype. So this is a field trial looking at the number of aphids landing on a plant for two varieties of brassica used, oilseed rape varieties, Samurai and Temple. And you can see that on Temple, there was a significant effect of the cisjasmone in actually reducing aphid landing behavior, but there wasn't for Samurai. So again, this reinforces this point that biopesticide activity is mediated by plant genotype. And I think that that's something that needs to be taken account of. And this is my final example of that, nearly finished. So thank you for staying with me. So this is another example, again, of the same BBSRC SARIC research program, whereby we're looking to see if plant genotype affects the efficacy of biopesticides. And this, again, this is for aphid control. This is with cabbage aphid, and this example is with two insect pathogenic fungi. So this is work done by Andy Gladman, who did his PhD with me here, and he's now working at ADAS and kind of continuing this IPM research at ADAS. So these insect pathogenic fungi, EPF, entomopathogenic fungi, they are used as biopesticides for glasshouse pests, including aphids. And remember, I said at the top of the talk, they consist of spores that are applied against a target pest. The spores germinate on the insect cuticle and grow inside and kill the target insect, in this case aphids. Okay, so when you're using EPF to control aphids, your target should be the nymph. It should not be the adults. You can spray, yeah, obvious against the adults, but the problem is it takes a few days for the fungus to kill. So by the time the aphid is dead, it's actually reproduced, and so you've got no controlling effect on the population. So you want to control the nymphs. They are your target stage, your target life stage. The problem is that the nymphs can molt, and when they molt, when they go from one instar to the other, they can shed the fungus on the cuticle, and so they can rid themselves from infection. So what Andy's work was doing was about using genotypes of brassicas that had partial resistance to aphids and slowed down their development. They didn't kill the aphids, they slowed down their development, and that gave more time for the fungus to actually germinate and penetrate on them. And that's shown in this graph here. So it's a bit complicated, but I'll take you through it. So it's a graph showing mortality of aphid nymphs. So it's equivalent to percentage. So it's going from 0 to 0.9, which is equivalent of 90% mortality. And we have two types of plant. We have a susceptible plant and a resistant, partially resistant genotype of brassica. This plant, it doesn't kill the aphids, it slows down their development. We have a mock, which is an untreated control here. We have two different types of fungus given by their codes. We've got this one, 172. This is a fungus called Acanthamyces, which is really good against aphids. It was actually developed here, and it used to be available commercially, but unfortunately it's no longer available registered for use. But it kills aphids really quickly because it germinates fast on their cuticle. And as you can see here, both the susceptible and the resistant genotypes, you get in good levels of control. This code here, 433, that's our code that we use for a fungus called Bavaria botanigard, which I've referred to previously in this talk. And we know that this fungus can kill and infect aphids, but it's not very good against nymphs because it takes a long time to germinate. And the nymphs, when it's germinating on them, they can, I said, they can molt and get rid of fungal inoculum. So this is for the susceptible genotype, and you're getting kind of 35% deaths of aphids. But when you spray the fungus on a partially resistant genotype, you're getting 70% mortality because the aphids are taking longer to develop, and so it gives more opportunity for the fungus to kill. I hope it's quite complicated, but I hope I've explained that properly. And I think this is really important for IPM going forward, is that we need to be combining plant breeding with biopesticides to actually understand the interactions within the system. In fact, we need to do more than that. This slide is about smart farming. So we need to be using the new advances we're getting in things like smart traps, which Rosemary works on, new advances in crop imaging for early detection of pests and diseases, using data analytics and new advances in smart AI and machine learning to really identify when we're getting the early stages of pest outbreaks. And we need to be using new advances in engineering to give us the precision application that we need for biopesticides, not just the timing of application, but the delivery of pesticide sprays to hard to reach parts of the plant, like the vertical surfaces and underneath the leaf surfaces. And all of that is possible. It requires novel thinking, and it requires engineering solutions, which is something we are starting to do here in our Agritech initiative at Warwick University. And that's it. This is my final slide. Sorry, as normal, I talk too much, so I've gone on a little bit longer than I was hoping. But this is my kind of take-home messages. So I think we need a kind of a roadmap for developing biopesticides, because they are increasingly part of the mainstream. we need to have effective systems for their use as part of IPM. And as part of that, we need to develop an ecosystem, by which I mean all of us who are stakeholders need to be working together, because we all want to deliver safe, effective plant protection products to farmers and growers as part of IPM. We need a more effective product discovery pipeline. We need to address issues around mass production, formulation and application. I focused on application today, but there's more to talk about in terms of mass production, formulation. We need innovation in regulations so we can get these safe new products in the hands of farmers and growers. As I said throughout this talk, we need to have IPM systems that capture the benefits of biopesticides and mitigate for the downsides. We need activities that are coordinated, that are funded, collaborative between research organisations like Warwick University and with industry. As part of that, we need a strategy for pesticide precision application. And as I said in that previous slide, we need to embed the use of our biopesticides in smart farming. And you know, this, and I know this is awful, this is kind of the boilerplate thing, but we're all in this together. You know, we want a sustainable food system, we all want healthy people, healthy planet, and a vibrant farm industry. And everything I've taught today is a small part of that. So I think hopefully everyone who's on this call, we're all working together in the same direction, which I think is really, really exciting. So these, I don't have time, aware of time. These are all the people that we worked together as a team to kind of deliver the research that we've talked about over the last 45-50 minutes. And that's it, Rosemary. I shall tour de force. I shall stop sharing my screen, if that's okay.
Brilliant. Well done, Dave. A really great overview. So you've got quite a few questions. So I think if you're happy, we'll go through them, and then I quite understand if people have to leave the call, but they'll be in the they'll be in the recording. So yes. Can you see them in the chat?
I'm just swiggling over to sorry, fiddling with that. Yeah, I can see that now.
So if you start at the top, which is just under the Gov UK logo.
Can you give me? Yeah, I'm just fiddling with my my screens at the moment. I have multiple screens, and I'm going to put them here, otherwise you'll just see the bad side of my face. Right, okay, so what was the first one then?
Is it good to use Sonata as an example, considering it's a withdrawn product and active wasn't supported for renewal either here or in EU?
It is good to use it because it's an illustration of proof of concept as a biopesticide. I do take your point, and yeah, I take that fully on board, but I was really using it as an illustration to show that published research is there in the public domain to show that biopesticides can give effective control. Right, so we've got next one here, Bavaria bar chart.
That's it.
Yeah, did not show response time and if safe to phyto numbers. So I think that was into the work that we did on tomato crops, where we were using the Bavaria, the Naturalis product, as a second line of defense to see if we could replace phenbutadiene oxide. So response time, do you mean speed of kill? So the speed of kill is, I'm trying to have a think, it's in the paper, but it's going to be, well, we're not other tomato work with another Bavaria product, we were getting an average survival time of 3 days, which is the fastest. So these things take kind of 3 days, 5 days, maybe 6 days to kill. So it is slower than a conventional chemical pesticide, and that's something you need to bear in mind. Effect on phyto, no, we showed that they don't in this study that the product we used did not infect phytomites. I didn't have space to squeeze it in. In the study itself, there was a reduction in phyto numbers, but that wasn't due to infection by the fungus. It was due to a decline in the number of spider mites that the phyto were using for food. So do you get the point? So because otherwise the phytomites are actually feeding on spider mites, and that allows the population to increase. What you can do with these crop protection systems, so using phyto or other predatory mites in protected crops, they're applied according to a trickle release system. So you're applying them repeatedly. So even if there was a slight negative effect of the biopesticide, you can then make up the numbers by adding more of the predator. And remember, the point about this is we're not necessarily trying to eliminate the pest population in this particular example, though it did work really, really well. What we were trying to do by using the biopesticide as a supplemental treatment is to just hold back the pest population to give time and space to allow the predator population to catch up and get back in control. Right. Okay, so how can naturalis be a second line of defense if its label clearly says apply at very first appearance of pest? You are correct. Absolutely right. And yeah, I don't know what to say. Yes, you are right. And that's an issue that we need to address. And it does say that clearly on the labels for these product. And yeah, yes, you all I can say is yes, you are right. And there are issues then about label recommendations, and there are issues about how we get the best out of these products. You know, to my knowledge, there's nothing to stop you using this product as a second line of defense, but at the moment, and there are people who are more qualified to talk about this than me, but at the moment the label recommendations and are all about effectively considering these products as standalone treatments rather than using IPM systems. And CRD will probably want to comment about that, but that's something we need to take forward. I think it is a very, very good point. Water volumes tend to be much lower in arable crops. Any thoughts on how this might affect efficacy of biopesticide in his crops and how this can be managed? From Sasha White. Yes, you are right. And actually, this is another important point in that there's a lot that different crop sectors can learn from each other. So arables in horticulture in this particular case. So in arables, you're going down to 60 litres per hectare. And I think horticulture can learn a lot. The idea of using a lower water volume is that in principle, you still get at lower water volume the same coverage on the crop, but you're getting more concentrated product on the leaf, and then that should translate into efficacy. There is a question, though, about whether the particular biopesticide you're spraying requires water for its activity. You know, if it's a biofungicide or a bioinsecticide that's a living microorganism, does it actually require water in order for it to germinate and grow? So there may well be, Claire and I have talked about this, you know, and we say, well, there may well be a minimum water volume that is required for activity, but that's something we need to take forward. Why is the number of applications per crop or season so limited for biopesticides, considering their much safer profile? Why are they treated the same as conventional pesticide when it comes to use of adjuvants, half rate or less? So you are right. Again, I'm probably not the best qualified person to talk about this, but you are right. There are limitations made on the number of applications. And logic tells you that if you have a product which does not have a negative impact on the environment and safe for people, then a priori you may be an argument for using it more frequently. And that's something, you're right, we need to capture and we need to take forward. The issue about adjuvants, I'm not quite sure what you mean by using adjuvants half rate or less. I would say, because I'm not an adjuvant expert, but I would say that we need to develop best biopesticide adjuvants. So we need new types of adjuvants that work specifically with biopesticides, both the living microorganisms and the natural products. That may well be a challenge because remember, as we move forward in crop protection, we're dealing with a wide range of entities, but that's certainly an area of work that needs to be taken forward. Are there any cis jasmine products on the market? No, not that I am aware, and I'm not sure what the registration status is of that particular product. And remember, that's not that what I'm saying doesn't just apply to cis jasmine. There are a whole range of potentially useful candidate biopesticides that have been developed through research, some of which are going through the authorization pipeline in the UK or in the EU as we speak, some of which have not entered the authorization pipeline, although there's been some really interesting kind of research done through them by universities and other research organizations. And that itself raises this question of how we actually get more effective regulation and authorization to deliver product to the growers' hands. And it's not just issues about having innovation in authorization and regulation, it's also about issues to do with niche markets and issues to do the fact that now a Brexit issue, that now the UK is no longer part of the EU, or Great Britain is no longer part of the EU. We are a less attractive market for plant protection products because we're a smaller market. So that's something we need to think about too. Do we know what the effects of control measures are, say for powdery mildew, by the application of UV light might have on biopesticides? So that's an interesting question, and I have spoken with people who have been using UV effectively for control of powdery mildew. For example, there are robotic systems, automated robotic systems now available in which robots go along and they apply UV light to strawberry crops, and that can be effective at reducing powdery mildew. But also you have to be aware that UV light could be damaging for biopesticides. It could also be damaging for natural enemies. And again, it's a very good point, and it's something that we need to be aware of. So I've spoken to a company who have been using UV light, and we both agreed, yeah, that is work that needs to be taken forward so we can work out how best to integrate UV with biopesticides and other components of the IPM system, which is the wider point, which was kind of really illustrated by Rosemary's excellent IPM pyramid, is that you have to understand whether there are interactions between the different components in the system. What have we got there? Excellent overview, Dave. You're amazing. Fantastic. Yeah. Okay, get that. Right. Well, with the reduction in funding to agriculture, how do you think SMEs will keep the funding for their research? Much of their fund comes from agricultural organisation growers that get PO. Yes, you were right. So this was part of the slide I did at the end about a roadmap and a strategy, and it also kind of relates to the government's national action plan as well. So I would like to see a call for arms for the government through DEFRA, because it's fine to have a national action plan and a set of targets, but you need to have mechanisms for delivery. And I know that money is tight, in not just in this country, but throughout Europe and around the world. But we do have to invest in our agricultural systems as part of the green transition. I don't think, I mean, my personal experience and feeling, that I don't think many politicians and policymakers are aware how vulnerable our food supply chains are, and the fact that we really need to invest more in sustainable food and plant production, including investment in the development in new types of crop protection agents. And that's really important. And you are right, yes, SMEs are dependent on public investment in order to develop their products through the technology readiness levels. And in fact, it's not just the SMEs, it's also the multinationals, because the large multinational companies, their business model depends on picking up interesting products developed by SMEs. So if we don't have public investment, if we don't have a mission economy, if we don't have the Mariana Mazzucato kind of system of using public money for investment, we won't get these new biopesticide products that we desperately need for crop protection. Sorry, I'll stop rant. I've gone into my rant, one of my rants, Rosemary. How can we access the SEALSO calculator? I will ask Claire, because it should be available on their website. So, but if you Google SEALSO spray applications, then you should see them. But I'll speak to Claire about it as well and make sure that's available. Can you recommend a biofungicide suitable for glasshouse use on brassica crops, specifically to control? No, I cannot. So I'm not a basis qualified person. I'm a university researcher, so I'm not in a position to recommend individual products, and that's not something that I will do. But what you need to do is to speak to, I don't know who where you come from, but you need to speak to an agronomist, or you need to contact the IBMA, the International Biocontrol Manufacturers Association. We should maybe make a link available for them, but and speak to them in terms of access to products. Comment from Martin about adjuvants. Adjuvants generally limit the amount of plant protection product to half rate or less, which seems a bit odd when it comes to biopesticides, where contact and coverage are crucial. Yes, I think that's a really interesting point, and we will capture that. I'd never thought about that before, Martin, but I think that's really useful. Thank you. Oh, Claire is listening, and she's saying not sure it's on our website. This is for the calculator. I'll try to make sure it is. Yes, so that's an action on Claire. She'll make that calculator available. And I think I'm a bit exhausted now, and I got my coffee and it's gone cold. So that's kind of annoying.
Well done,
yeah.
Dave. I think you worked really hard for an hour and a quarter. So yeah, brilliant.
I've earned an extra coffee. Yeah, well,
thank you very much.
Sorry we've gone on. I didn't intend. I always do talk too much, but it's a really interesting topic. You know, generally, like the webinars we're covering on this series, they're really valued. There's lots of food for thought, and I think the key thing is we all need to work together to take these things forward, you know, as part of a broader ecosystem. So thank you, Rosemary, for inviting me, and I hope people who listen actually found this useful. Right, and I will say goodbye.
Brilliant. Thank you very much, Dave.